Throughout the genetics course that I teach, we begin with the Mendelian form of inheritance and then go into the exceptions to Mendelian inheritance. Later in the course, how chromosomes and their defects can affect chromosomal inheritance. So different forms of inheritance are mentioned throughout. And then as the course ends, a couple final topics are raised which hadn't come up not centrally before. So first off, one non-Mendelian type of inheritance is when the cytoplasm of the ovum determines a trait. See, sperm are essentially DNA with a tail, so there's not much cytoplasm there. And the ovum is so large. I mean, for example, in humans, it is this cytoplasm, which then gets divided for the first week of the embryo's life. There's no nutrients coming from the placenta until a week after conception. So the ovum, has so much stuff in it. And so then the mother's genotype determined what was put into the cytoplasm of the ovum. But if there's two different alleles, that doesn't necessarily have to match the specific form of a gene that ovum has, because ova are haploid. They get half the number of the chromosomes. But some of the things in the cytoplasm could be determined by the other allele that different ova receive, but not one in particular. So it is known that in insects like fruit flies, you can look at, say, Drosophila larvae, or say caterpillars in butterflies, and say, that trait was determined not by a gene that is in the nucleus of the ovum, but rather by something that was in the cytoplasm. So two easy examples. One example is that if pigment is put into the ovum, that could determine the pigment of, say, a caterpillar. So it's not the genes that are in the nuclei of the caterpillar, but what was originally in the ovum. A second example is the organization of the cytoskeleton of the ovum could then ultimately affect how the embryonic cells divide and affect, say, the coiling of a shell in a snail. Once again, if you ask what's causing this, it's not a gene that's in the nucleus of the snail individual's cells, but rather something that was set forth by the cytoplasm of the ovum. And so this then is a non-Mendelian form of inheritance. In humans, the big example of this is in the mitochondria, which I'll get to, but I prefer to just mention one other thing before getting to mitochondria. With epigenetics, if you recall, there are ways that you can change either a nucleotide or maybe the proteins called histones around which the DNA is wrapped that will affect the expression of a gene, sometimes for long periods of time, sometimes even like into the next generation. This is done without changing the nucleotide sequence, but rather by these epigenetic changes. So for example, one is that a methyl group could be attached to the cytosine nucleotides. And so especially if it's in what's called a CPG island, where there's lots of cytosine and guanines, the more methyl groups you add, the more likely a gene is to be off. So here you have two identical sequences, but one will be used, this one will be used. It's an active gene making transcript, which makes protein perhaps, whereas this one is inactive. Why? It's not any nucleotide sequence. It's the methyl groups which are attached to the cytosines, which then determine that. Well, an interesting thing about epigenetics is there is an enzyme which will make sure that when a cell replicates, the level of methylation which was present in the original DNA before replication then gets copied, which means that whenever methylation is applied, it can become quasi-permanent until it's changed, because it gets replicated each time the cell divides. And interestingly, the DNA from sperm has a higher degree of methylation than the DNA from an ovum. So many cells would find themselves in a situation like this, that there's, say, 2 copies of a chromosome. So there's 2 copies of a gene, and there's the gene you got, from your mother and the gene you got from your father, and the cell can tell the difference because the cell that was inherited from the parent, the paternal parent, the father, has more methyl groups attached to cytosines. And every time the cell divides, it replicates that. So years and years later, the cell knows this is the copy I got from my mother, this is the copy I got from my father. And there are scenarios where a cell will preferentially express one or the other. So for example, let's say that one looks at the gene whose deletion can result in either Angelman syndrome or Prader-Willi syndrome, which vary. They can affect disposition, they can affect erratic movements, they can affect weight gain, even perfect pitch with music. And so if the mother's gene is deleted, then the result in the individual is called the Angelman syndrome. But if the paternal gene is deleted, then the result is called the Prader-Willi syndrome. So the same section of DNA could be deleted, but the effects depend on the parent. So this phenomenon is known as imprinting, where a cell could express one parent's gene or the other, but not both, or express them differently. So a cell can tell the difference between the form of a gene one got from one's mother and father and can express them differently. So if an individual develops Wilms tumor, all right, there will be consequences in phenotype. But which consequences depend on whether the mutation came from the mother or the father. In mice, for example, the insulin-like growth factor, IGF, mice only express the male copy, but the receptor mice only express the female copy. And so there are a number of human genes like that as well, where our cells can tell the difference between the form we got from our mother and father and express only one. So once again, this is what is known as imprinting. Now, the best example of non-maternal or non-Mendelian inheritance and maternal inheritance in humans involves mitochondria. Now, mitochondria are absolutely fascinating. And I'm, often disappointed because, as biology courses try to cram a lot of material in, a lot of students will learn the mitochondria is the powerhouse of the cell and go on. Yes, it's true that, your foodstuffs are broken down here and ATP energy is produced. But mitochondria do much more than that. And they're fascinating. There's so much more to them. So one of the things about them, which is fascinating, is they have DNA. So there are 2 parts of your cell which have DNA. The nucleus, which has those 46 nuclear chromosomes. Remember, we talk about humans have 46 chromosomes. But then mitochondria have their own chromosome, all right? Now, this is the cells of a turtle's liver. You can see these big nuclei. See all the little dots? Those are mitochondria. A cell could have 10 mitochondria. It could have 10,000. So the mitochondrial chromosome is small, but if you have 10,000 copies, that then nevertheless is significant. So, and I tell my students, it's actually not correct. Like I've told you before, you have 46 chromosomes. You have 46 chromosomes in your nucleus. You have another one in your mitochondria. And if you've got 10,000 mitochondria in a cell, that's an awful lot of DNA that is different from just then the nuclear genome. Now, this can have lots of effects. I'll go specifically into the DNA in a second. But mitochondria are not just involved in the second and third steps of cellular respiration, but there's also these membranes from the endoplasmic reticulum that attach to them. So you could refer to them as MAMs, mitochondria-associated membranes. 20% of a mitochondria could be covered by these specialized membranes. And there's, special exchanges going on there. The mitochondrial associated membranes and the regions of the mitochondria are important in the calcium levels of the cell, and the calcium is important in intracellular signaling. When we talk about cancer, mitochondria are extremely important in programmed cell death, what's called apoptosis. So if a cell begins to behave badly, it can self-destruct. All right, cells can be told to kill themselves so that they don't become cancerous, and mitochondria are crucial in that. And so obviously anything that affects mitochondria can have lots of effects in the cells, because mitochondria are doing more than just breaking down foodstuffs. This chromosome in humans is composed of 17,000 base pairs, although mitochondria can vary and in size. So some organisms have mitochondrial chromosomes which are bigger than others. There are more than 100 diseases which have been linked to mutations not in the DNA of the nucleus, but rather the DNA of the mitochondria. So this DNA can have mutations, and it can cause a number of diseases, and I give a few here. So very often these mitochondrial diseases, are in very active cells, and so active cells then need, lots of mitochondria. There are specific genes here, and so there are, I'm sorry, I got past it. So there aren't that many genes located here. So 13 proteins are encoded by the mitochondrial chromosome, in addition to 22 tRNAs and 2 rRNAs. And so this is a small chromosome and compact. The reason that I'm discussing it here is not only that it is involved in inheritance. There's DNA which has genes, and mutations can cause disease. But when one considers then the moment of fertilization, there are mitochondria in the sperm, not many, but there are, but typically that's in the mid piece of the sperm. And then that typically does not enter into the ovum, just the nucleus. And so while there is DNA in the mitochondria of the father's gamete, that DNA will not then contribute to the embryo. So it is the mitochondria of the mother in the ovum, which will then be the ancestors of all of the mitochondria in an individual later in life, so that every time cells divide, these mitochondria have to divide by fission. So the nucleus can't encode more mitochondria. The mitochondria just have to reproduce themselves. And so thus the existing mitochondria are only of maternal origin. So if you inherit, and you have obviously, mitochondrial chromosomes, all of them came from your mother. So notice that is a different type of inheritance. It's not sex-linked inheritance where there's an X and a Y. No, it's just maternal origin. Now, the ovum can then have different types of mitochondria. So there could be mutations in some of the mitochondria and not others. This is known as heteroplasmy. And so therefore the inheritance of mitochondrial chromosomes can be a little complex in that way. And when I said like there could be specific diseases linked to mitochondrial mutations, also things like cancer. So some individuals, cancer is caused primarily by a nuclear gene, but in other cases, a mitochondrial mutation has a major role. In other ones, it might play a minor role, so that multiple mutations are contributing to cancer, some of which are coming from the mitochondria. But notice that, here you see sex-linked traits, X's and Y's, but this is determined by the nuclear chromosomes. All of these individuals are getting mitochondrial chromosomes from the mitochondria, but just from their mothers. So that's another type of inheritance different from, those other forms discussed. And so the reason for this is that if we ask, where did the eukaryotic cell come from? Right, so all eukaryotic cells have either mitochondria, or in the case of something primitive like giardia, something that seems to be a relative of the ancestors of mitochondria. Well, the model, which has a great deal of evidence, is that endosymbiotic origin of mitochondria and chloroplasts. So if you were to say, well, let's look at mitochondria, or in, say, a green alga, the chloroplasts, they look an awful lot like this bacteria, a proteobacterium, and then this blue-green alga, this cyanobacterium, all right? And when I say they look like them, their size and shape are the same. As I had mentioned, the mitochondria and chloroplasts reproduce by fission, just like bacteria do. The nucleus can't make more. These chromosomes that they are circular chromosomes, which is odd because circular chromosomes are typical in bacteria, but the chromosomes in the nucleus are rod-shaped. So you have two completely different kinds of chromosomes. One looks like bacterial chromosomes in your mitochondria. The rod-shaped ones are in your nucleus. If you were to look at the codons which are used, like this 3-nucleotide codon codes for this amino acid, or a stop codon, or how translation is started, mitochondria do it the way that bacteria do, not the way that the rest of your cell does. If you look at antibiotics, the antibiotics that affect bacteria also affect mitochondria. That's why antibiotics in high doses are toxic to any of us. If you're allergic to an antibiotic like penicillin, it's typically because your mitochondria are variant and look a little bit more like bacteria than most people's mitochondria do. And so then you add in, there's other things which we find in bacteria, but also in mitochondria, like plasmids, the structure of the ribosome subunits, and specific proteins. And so the way the eukaryotic cell was born was not only the origin of the nucleus, but the origin of mitochondria and later chloroplasts, which came from endosymbiosis of bacterial cells living inside larger cells in a scenario where they both benefited. And your bacterial chromosome is derived from your mitochondrial chromosome is derived from the bacterial chromosome of those first endosymbionts. All right, so so obviously that's fascinating, but once again, another example of a different type of inheritance.